I still remain Mrs. Usman Halima, also from the Department of Geology and Mining. You are still welcome to this week's presentation for week 4 and 5 on invertebrate paleontology. Today we will be treating the phylum Porifera, its characteristics and classification. Sponges, or simply referred to as Porifera, simply referred to as sponges, they are named or derived from two Greek or two Latin words, porous meaning pore and fera meaning bear, that is their pore bearing. The bear pore simply means an opening to which either respiration, feeding, or probably reproduction, a means of passage that comes in and out of an animal. Sponges are simple invertebrate animals that live in aquatic habitat. They belong to the phylum Porifamily called phylum Porifera. They are the lowest multicellular members of the kingdom Animalia and contain over 500 species. The members of this phylum are referred to as Porifera, as we earlier stated, and have pores known as the osteas. On the beginning, we say they contain pore or they are pore bearing. These pores are referred to as the ostia and are found all over their body. The sponge-like bodies have the ability to absorb and withhold fluids. In the last, maybe one of the videos I was probably watching, it was derived that there is this category which we refer to as this sponge, probably is derived from that name, or just the sponge. Maybe an image will describe it better. It actually looked like one of these plants, which mostly in our rural areas we try to use it actually as sponge with a plant just like cucumber. When it's dried, you see this spongy this thing is used either to wash plate or even to take a bath. This is just the best description I can describe how this type of animal look like. These creatures remain attached to the substratum and do not move. That means they remain in one place and they are not moving. Because of this, they are regarded as plants. However, the life cycle and feeding mechanism discovered later proved that they belong to the animal kingdom. So it is from the early studies postulated that they are actually plants, but previous or more recent researchers were able to describe them as animals. The characteristic of this phylum Arthropoda are as follows. We say first, the members of this phylum are commonly known as sponges. We've earlier stated that Poriferum bear numerous pore spaces known as pores known as ostia, and we say this found all over their body, that is in the outside, or what we refer to as the cell wall, the body wall, which leads into the central cavity known as the spongiocele. The spongiocele opens to the outside by an osculum. So all this we are going to see it in the morphology or a typical description of what a sponge or a porifera look like. They are most primitive multicellular animals that show cellular level of organization. The cells are not organized into tissue and are arranged as loose aggregates. They are mostly marine and a few are freshwater forms. They are often asymmetrical and the body is irregular, cylindrical, or vase-like. Unlike the first phylum we treated, we said they are bilaterally symmetrical. This one we say they are asymmetrical and the body is irregular, but we'll still eat as we move on. 6. The body is supported by a needle-like structure known as the spicule, made up of cilia or the spongy fiber. We'll still take a look at it. They are diploblastic animal and contain an outer dermal layer of pinacocyte and an intergastrial layer of choanocyte. This group of animals they are just as say they possess a cellular mode of organization, and then we have different cells that perform different functions in this organism. Just to mention a few, are the pinacocyte, which is at the cell that are covering, or the outside coverage of this organism has these cells known as the pinacocyte. That means they are the external or the wall covering. The choanocytes are probably the ones inside, or the digestive cell, which is used for digestion, the digestive cell which is used for digestion. The nervous and sensory cells are probably not differentiated. They also possess a canal system which we refer to as a canal system in porifera, and it is system of interconnected canals through which any other means, be it food, be it metabolic activity, or maybe reproduction. This is the canal where it passes, and then we have basically or the grading of this canal system, which we see in the as we go. They are mostly the asconoid, the synconoid, and then the leuconoid. All these canal system are described from a simple to complex, and then to the most complex one, be it the simplest, the ascon, the syncon, which is the complex or the intermediate, and then we have the leuconoid sponge, which is the most complex. This is just a label diagram of a sponge. We have the osculum, which we described earlier, that the water enters through the ostia and then out through the osculum. This is the porocyte, which is the pores where water enters and then leaves. We have the spicules, the spongocyte, which is the inner coverage, the choanocyte, which is the digestive or the digestive cells, and then we have the epidermis, or where the pinacocytes line the outside covering of the line the body wall of these animals. Then we have the types of the canal system, which I earlier explained. We have the asconoid or the ascon canal system. This is a very simple one, in the sense that the water enters through the ostia and it goes out through the osculum. The syncon sponge, the water enters through the ostia, it goes into some of these cells, it enters just as it is illustrated in the figure, in the image. It enters into some parts of this, be it the choanocyte, the spongocyte, and so on and so forth, and then goes out through the ostia. The leuconoid sponge, which is the more complex, the water enters through the ostia. It goes into so many tiny numerous cells, run into the body, and then it comes back into these such cells, and then all is collected by one osculum, by collected and then excreted, or goes out of one of the canals, one of the tissue, or one of the cells referred to as the osculum. Types of canal system in sponges. In sponges, the canal system is of three types. We've earlier discussed this: the asconoid canal system, the synconoid, and then the leuconoid. Just to lay a more emphasis, which is this is the simplest with thin wall. Spongocyte is large. Choanocyte and gastral layer line the spongocyte. So this is a thick wall, is narrow, and then restricted to radial canal. This is the most complex one because it's folded thick. Choanocyte are confined, and flagellated chambers are formed by invagination and radial canal. Digestion is intracellular. Respiration and excretion are through the body wall by diffusion. Reproduction: to this type of animal exhibit both sexual and asexual reproduction. Asexual reproduction mostly simply mean reproduction by this vegetative part, as we are taught in the lower level. Then when we are in school, when we are in the secondary school, the vegetative parts of these are used for reproduction and is referred to as the asexual reproduction, or it's occurred by, in this class we try to describe it that it occur during fragmentation, budding, and then the gamete. Sexual reproduction in these sponges occur when gametes are generated. This is just a diagram to illustrate how the sexual reproduction and then the asexual reproduction takes place. We have the asexual reproduction through the vegetative part. We can see the arrow towards our left, how it is being cut, and then it's being cut, deposited on the ground, it generates to form a sponge. On the other side, towards the right, we have the sexual reproduction, where we have things that look just like the sperm cells, and then it's deposited into, or maybe into the female organ, what we refer to as the female organ, and then from there gametes are formed and a new sponge develops. Classification of phylum Porifera. Porifera are classified into three classes as follows. One, we have the class Calcarea. We have basically trees, the Calcarea, the Hexactinellida, and then the Demospongia. The Calcarea, I would say they are mostly found in the marine, shallow water, and coastal habitat. Skeleton is made up of calcareous spicles. Spicles are just these are just part of the body that look just like the needle. The body is cylindrical in structure and exhibit radial symmetry. Large ionocytes are present. Small size species are present in this class. Example are the Skypher and then the Claritna. The second class is the class Hexactinellida. They are also found in marine and also in deep marine. The skeleton is made of siliceous spicles. Unlike the class Calcarea, that is made up of spicles, but these spicles here, they are siliceous. The body is cylindrical in structure and exhibit radial symmetry. Small cyanocytes are present. Moderately sized species are present in this class. An example is the Hyonenemena and the Euplectilla. Class Demospongia, it also contains the largest group of these sponge species found in marine and freshwater. Skeleton is made up of spongy fibers, cylindrical, and exhibit asymmetrical body structure. Very small cyanocytes present. Large size species are present in this class. Example is the Spongilia and then the Spongia. Poriferan cells, just as we discussed earlier, we're going to treat some of these. We've treated the part of the characteristic, the canal system. Another thing is this: we're going to look into these sponge cells and then probably discuss their functions. We, as well as said, this type of this group of animals do not possess a structure or an organ. For instance, sponges do not have maybe like stomachs, kidney, lungs, and so on and so forth, or maybe liver and so on and so forth. Therefore, what they have these cells we refer to as body cells for their day-to-day activities in order to sustain their life. These cells can be viewed, or a microscopic view of these poriferan cells is shown on the slide on the screen. You have the spicle, the archocyte, the sclerocyte, the mesenchyme, which is just a thin wall in between the pinacocyte and then the choanocyte, which is the digestive cells. We have the porocyte, and then we have the osculum or the ostia, where the water enters, and then the outflow, which is the canal system. We have the flagella, the column. So this in this image, you can see the interior and then the exterior part of these cells, how it is located. So whatsoever it is, the movement of all these water, nutrients, and so on and so forth is through the help of flagella. Because you can see there's an arrow pointing to both sides, left and right. From a particular point is the interior, which is the inner part, or when you cut a sponge, that is what probably you'll be seeing inside, and then the exterior is the microscopic view, is what is helping us to see what and what is contained in the microscopic view of this animal. Pinacocyte, as we mentioned earlier, is the skin cells of these sponges. They line the exterior of the sponge body wall. They are thin, leathery, and tightly packed. Choanocyte, these are distinctive cells which line the interior part of the body. The cells are a central flagellum that is surrounded by a collar of microvilli. It is strikingly resemblance to a single-cell proteus called choanoflagellates that makes many scientists believe that choanoflagellates are the sister group of this animal. Definitely, in terms of evolution, we have this animal from this stage to the other, and then they try to develop adaptive features, which makes some of the presently or existing forms looks like some of the extinct form of these animals that we study in paleontology. Choanocytes are versatile. They flagellate beats and create active pumping through the sponge, while the collar of the choana are the primary areas through which the nutrients absorb into the sponge. This is the sponge cell that develop into gametes. Mesenchyme, as I mentioned during this plate of the microscopic view of that view of the sponge, between these two layers is the mesenchyme. The mesenchyme consists of a proteinous matrix, some cells, and spicles. Archocytes are very important in the functioning of day-to-day activity. They are totipotent, which simply means they can perform functions of other sponge cells. as they can ingest food, they serve to transport nutrients or water or materials, and they can also develop into gametes. That is, we can almost say that the choanocyte, a compound form, or in summary, is almost a working engine of this animal in case the other cells failed. The sclerocyte is the secretion of spicules is carried out by this cell. Other cell made up of the spongiocele secrete the spongiocele skeletal fiber where these are present. We have the myocytes and then the porocyte. We said the poroferans do not have any muscles, so their movement is limited. However, this porocyte and the myocytes they contract in a similar function as the muscles, which surround the canal opening, and pores can contract to regulate flow through the sponge. This is an assignment for this class, and then we look forward, as I used to state in each of the class or each of the contact, there are needs for update. I will still update and then keep the students posted. And then this is an assignment for this class. Thank you for listening.